That's okay.
Thank you so much.
Mm-hmm.
And Judy, do you want to meet after this still?
Yeah, we can. I'm wondering why the slides are now stuck. Hmm. Hmm. I'll just do it manually. I want to make sure to mention there we go, the meetings, Keystone meetings.
Yeah, you're good to put those up already. I can tweet too. I just
That'd be great.
Yeah.
I stopped tweeting, but
Oh, you did. Okay.
but I
We should get a Blue Sky
yeah,
account too for the Myeloid network.
yeah. And
Did you move to Blue Sky or?
I haven't done it yet, but I will, I think.
I
Although,
created an account. I'm not very good at it. It seems much more cumbersome to
oh,
like
yeah.
tag people and things, but I'm sure I'll figure it out.
And I'll post this on LinkedIn. I don't know if
Oh,
I
good.
set up a Myeloid Network LinkedIn page,
Yeah,
maybe.
we should. We should.
Great.
Yeah.
Are you going to any of the Keystone meetings or speaking at them?
This year, no. The 2026 Myeloid one, yes.
Well,
And
you're the chair, right?
no, I'm the chair of the tumor immunology, which is the month after. So I'll do it in 2026. But sometimes I find I'm invited to Keystone a little bit closer to the meetings, maybe when they need someone to fill in. Still a fill-in.
No, not if you're a chair. You're not a fill-in.
Well, yeah, in 2026.
Well,
Hi,
I'm
Simon.
hoping they will positively respond to our meeting
Yeah.
plan.
Yeah, thanks for putting that together.
Yeah.
Hi, Simon. How are you?
Not bad, how are you lot?
Hi.
Good to see you. Thanks
Thanks
for
for
speaking.
That's all right. I've had my instructions from the boss.
Who's that? Dimitri?
Yeah.
Hmm.
Don't know if he's here yet.
I know how you feel. No, I'm just kidding.
I'm joking. Ask him about the snow.
Yeah. He should be on soon.
Hi, Judy. Hi, Jennifer. Happy New Year.
Hi, Jelani.
Hi, Jelani. Happy New Year.
Judith, what happened to Ganalisib? Did it just evaporate?
So Infinity ran out of money and then
Yeah.
they sold their assets to Deerfield, which set up a company to develop it for myeloid leukemia. And that right now they're not planning to immediately do trials in other diseases, but I've been trying to convince them to do it. So they might work with us if we can fund the trials. But I don't know if you saw there are 3 papers on PI3 kinase gamma driving myeloid leukemia, AML, and
Yeah.
Andy
Dimitri said
Lane.
those. Yeah, they were recent, right?
Yeah, and Andy Lane from Harvard. I think he's the founder, maybe, of this company.
Yeah. And he's going to present at the Myeloid Network in the spring, right? We have to
No, he couldn't do it in March, so we have
we
to
couldn't do it in March, maybe this summer. Okay.
Oh, that'd be cool.
Yeah.
Yeah. Are they ever going to publish the clinical data then? I guess no, nobody's got it.
Well, we have biomarker data from clinical trials that we currently have under review, and we
that's good.
published some biomarker data from the phase 2 breast cancer trial in JITC. And as far as the results go, it's not people are working on it, but it's excruciatingly slow. So
Hmm.
I think that the clinical investigators are going to publish it, but it took a long time to publish the phase 1 data too.
Yeah, because I was just looking to see whether the clinical trials had been published, because I think that data is still the most pivotal data for the macrophage myeloid field.
I agree completely. And I can talk to the only contact left is Brenda O'Connell. I'll ask her what's going on.
Yeah, it's such a shame, really.
Oh, I agree.
Yeah, I mean, obviously.
Simon, is there a gamma inhibitor in development at AZ?
We do have a gamma inhibitor, which I'll talk about a little bit today, but it's not in clinical development. It's on hold,
Mhm.
and actually, you know, yeah.
So there was a paper based on one of your AZ compounds that made it into a PROTAC, and they published that for AML.
Oh, which
So
one?
we'll see what happens.
On the gamma one?
AZ2? I don't know what that is actually. Do you know?
No.
Yeah. But
On a gamma delta or a gamma?
they claimed it was gamma. But
Yeah. I mean, where they always have to be a bit careful is how much they're dosing up to delta for the AML indication,
I know. Yeah.
and then they're going to end up in the same pickle that people were in with the delta inhibitors with the FDA.
Yeah. I
Yeah.
agree. But we haven't given up. We're working on some inhibitors.
Well, what would be really, I think, really, really good is for the clinical data from 549 to be in some peer-reviewed journals. I think that's going to make a
Great.
massive difference. Here's the boss.
Hello.
Hello.
Great. Just finish another meeting. It's always like that. Okay.
Mhm.
You can hear me well?
Yes.
Yeah.
All right. We have 60 people now, so we'll give another couple of minutes, right?
Yeah. And then I'll just mention these Keystone meetings. Let's see.
Is anyone going to the Keystone in Banff?
Which
I don't
meeting
know
is
why
it?
they want to go to this place. Yeah.
It's a nice place.
Jelani, what meeting is that?
I-
Is that one of the ones that was just advertised?
No.
Wasn't one of the ones that it's next week. It's in Banff, Canada. It's a tumor microenvironment meeting.
Oh,
Mm.
It's a lot of good meetings.
Yeah.
Thanks for that.
Yeah.
That's made me feel really like miserable. Unlike Dimitri, I love Banff.
I do
Banff
too.
is a beautiful
I don't
place.
care. I think that we have way too many meetings. People going from one meeting to
Oh, I'll be seeing you
another.
later
and
then. I'll follow.
I don't know. I mean, how much information you can get out of this.
I was thinking more about Banff actually, Dimitri.
Yeah,
Yeah, well, yeah. Yes, that's true. That's true.
All right, I'll just say a few words about the Myeloid Network. I want to welcome everyone back to the Myeloid Network in 2025. As a reminder, we were founded in 2020 to advance knowledge about myeloid cells and myeloid therapy. And we want to especially give a shout out to meetings that are being put on by the Keystone Symposia that you see here that would be of interest to our community. And we welcome Dmitry Gavrilovich to introduce today's speaker.
Yes. Okay. Welcome, everyone. We have very interesting day today. We have more practical assessment of whatever we discussed so many years on a theoretical plan, and the best person who can do it it's Simon Barry. He spent almost 25 years in AstraZeneca, growing in ranks, and now he's executive director running basically biomedical research in small molecules targeting different population different cells, different projects from AKT signaling, from ATR inhibitors, and many others. However, his interest for many years was and still is in myeloid cell targeting. He was at the beginning and in developing the whole slew of myeloid targets. And he published extensively. Basically, I'm pretty sure quite a few on this call either got molecule from him or collaborate. What he will bring us is absolutely unique perspective on how those enormous effort people expend to develop drugs, how it translates into actual clinical data. What's wrong with this? What's good? What's working? What is most likely not? And we really need kind of different perspective on what myeloid cell investigation and targeting is. And Simon, with his fantastic contribution to the field, will be the best person to deliver that. So Simon, thank you very much for coming, and I'm looking forward to your presentation.
Thanks, Dimitri, and thanks for the invitation to talk. Apologize in advance for anybody who's seen elements of this before, but what I'm going to try and do is go through a partially historical perspective on projects that we've run in AstraZeneca all the way to candidate drug and beyond, to look at different elements of myeloid biology as a resistance feature, primarily to IO. And then I'll talk about what have we learned from those, and despite the fact that we still yet to have a registered myeloid therapy in the oncology field in the clinic, the fact that all of this learning actually gives us some really good insights into how to make a success, either from mechanisms that we've already tried and have not progressed as far as we believe they should, or for the new types of targets that everybody presumably on the call are identifying with their own individual research. Oh, disclosure, evidently I'm a AstraZeneca employee, as Dimitri pointed out. So approaches to target myeloid cells. I mean, everybody be very familiar with the types of mechanisms that we've highlighted on the left. This is a review that Dimitri and I wrote that came out last year or the year before. Different ways in which we've, as a field, taken the therapeutic project forward is to both deplete or inhibit the recruitment of both neutrophils and PMMDSCs or macrophages, monocytes, and MMDSCs to the tumor microenvironment. And then more recently, with the work that Julia's done and other projects, look at agents that can also reprogram how these cells are arriving at the tumor microenvironment. So look at ways in which they can remodulate the whole tumor microenvironment, not just the suppressor function, but the whole tumor microenvironment, to enhance either chemotherapy response, which is where I originally started with my interest in myeloid cell resistance. It was actually, for me, it was VEGF and chemotherapy, not immunotherapy, which was many years ago, last century. And then subsequently to look at how these cells are regulating the full architecture of the tumor microenvironment. So from an AZ perspective, we had quite, and still have in development, a big portfolio of assets where we've taken different approaches to target myeloid cells, mechanisms that we think are important in driving myeloid cell regulation. So in the early phase, which I'm not going to talk about, we've looked at IL-8 versus CXCR2, which is an axis that we obviously all know and love as well, important for neutrophil biology. We've looked very heavily at the regulators of macrophage recruitment, MCP, CCR2, and CSF1R. And then we've also looked at other mechanisms like gamma and STAT3, PI3 kinase gamma and STAT3 that we know influence the differentiation status. And a program I'm not going to talk about today, CXCR4, we've also looked at that. Perhaps it's an agent that we didn't really progress as far as we would, but obviously there was exciting data with AMD 3100, where they showed clear modulation of the TME in pancreatic cancer. But today I'm going to talk about experience that we've got from our PI3 kinase gamma program with a molecule that is very close to meganelisib, which is obviously the molecule that Judith's been supporting for many years. Our CSF1 selective, CSF1R selective small molecule inhibitor, an arginase inhibitor, just to highlight some of the learning that we have within the immunometabolism space, and then CXCR2 and STAT3, which are two agents that we have taken to reasonably advanced clinical trials. So one thing I just wanted to highlight is that although unsuccessful, there's been a phenomenal amount of work on various flavors of molecules, antibodies and small molecules to inhibit CSF1R, CSF1, CCR2 and the CXCR2 IL-8 axis. And actually the composite of all of this information gives us a really useful platform for understanding how to construct our preclinical data sets to inform more successful clinical trials, but also to understand how the clinical trials have progressed, yeah, and to learn from them to refine how we approach things that we do in the future for new programs coming forward. Yeah, so although in the main these programs have not been successful in delivering registered products, apart from obviously the CSF1R, CSF1 inhibitors registered in GDCT, yeah, these are all really important molecules that we need to learn from. And one of the things that we have built an understanding of is how these different mechanisms that we've targeted in the clinic have reciprocal and cross-regulatory functions within the tumor microenvironment. So they're not just modulating T-cell suppression, but they're influencing a whole slew of biology that we're now learning more recently is different when you look at it from a disease specific perspective. So if you look at glioma, all the work that Joyce has been doing, versus pancreas, versus colon, even metastatic colon versus primary colon myeloid biology and influence, it's really clear from the emerging data that we now have that you can't use these as a one mechanism fits all diseases, but actually we need to look at them from a disease specific perspective. So the first thing I'm going to talk through is the work that we've done on CXCR2 inhibiting neutrophil recruitment to tumors. So this is where we started in the myeloid field within oncology and AstraZeneca some time ago, just around 2002, I guess. CXCR2 is interested to people working in many disease areas because of the pivotal role it plays in recruiting neutrophils to any disease organ. So whether it's asthma, COPD, or cancer, yeah, CXCR2 is a really important mechanism for controlling neutrophil recruitment. There are other functions ascribed to CXCR2 biology, such as the regulation of angiogenesis, and also the role that it plays in stressed epithelial cells to drive, for example, chemoresistance. But the biology that we felt is most dominant is the role that it plays in regulating neutrophil biology, although there is some caveats in prostate cancer. One of the pieces of work that we did early on in the CXCR2 program was to use standard syngeneic models to validate how CXCR2 was delivering benefit in an oncology setting. And we did the classical stuff, using CXCR2 whole body knockouts and small molecules, and to some extent neutrophil depleting antibodies. What this really taught us was that the simple syngeneic models for this piece of CXCR2 biology was not really giving us the type of data that we felt was discriminating and allowed us to really articulate how the biology was working in the context of a disease tumor microenvironment. So very early on, we initiated a collaboration with Owen Sampson's group at the Beatson Institute in Scotland, who have a whole suite of genetically engineered mouse models of many different cancers. So liver, colon, pancreas has been the focus. And what we wanted to do there was to use our assets to look more specifically at the role that CXCR2 may play both in primary disease and metastatic disease in tumors that were driven by oncogenes and were developed in an orthogonal tissue-specific manner. So the first study that was really informative to us was that when we started to look at the role that CXCR2 small molecules had in combination with gemcitabine in the KPC model, so standard KPC, but we looked at it in two different ways. One in an early disease setting, where the tumors were effectively restricted to the pancreas, and then in a second setting where we were taking them a little bit later and more morbid, where we knew that they would be developing metastatic disease. And what this revealed was two things. So one, that CXCR2 small molecules shown in the left had the potential to prime responses to gemcitabine. So we didn't use platinum-based chemotherapy in these studies. We used gemcitabine because that was vogue at the time. So you can see that there's a modest impact on increasing the efficacy of gemcitabine when you've depleted the tumor microenvironment of neutrophils. But on the right, you can see that also there was a big drop in metastasis to the liver when you combined CXCR2 inhibitors as a precursor of AZD5069, and which gave us a sort of insight that actually, 10 years ago or so, that the biology wasn't going to be as simple as the biology that we were deriving from the syngeneic models. Within this work, we then went on to look at the role that it had with the immune system. So at the time, this was the pivot between chemotherapy and checkpoint inhibitors. And what we found was that, again, CXCR2 combinations with PD-1 at the time could enhance response to checkpoint inhibitors. But you can see here in the bottom that this was only really given durable benefit in a subset of patients, and actually probably in reality mimicked what we would expect to see in any clinical trial, or maybe even overpredict what you might expect to see in any clinical trial in pancreatic cancer. But this was just a doublet CXCR2 combination. And this had the right biology, that it was associated with an influx of T cells into the tumor and into the tumor microenvironment, and they did appear to be in a more activated state. Using that principle of different results in the primary tumor and the metastatic tumor, and we then went on to look at the same biology in colon cancer. So what Owen had developed at the time was some models of primary colon cancer and this new derivative, this KPM model, KRAS P53 Notch model, where for the first time they were able to model in situ metastasis to the liver. These models align to very specific subsets of disease. And the KPM model that I'm showing here aligns to the CMS4 subsets. This is, if you can remember this will come back later. Yeah, so we looked quite hard at the role of CXCR2 inhibitors across models representative of each of these subsets. And where we found that CXCR2 inhibitors in colon really had a big impact was in, again, reducing the incidence of metastasis to the liver. What they weren't doing is doing anything to influence the primary tumor development. They were reducing metastasis specifically in this KPN model, which aligns to the CMS4 subset of disease. So that provided us with another piece of information that actually probably CXCR2 is important in the liver microenvironment rather than the primary tumor microenvironment in the colon. In parallel, we were working with Andrea Alamonti over in Italy, Bellanova, who was interested in the role that CXCR2 biology played in P10 prostate cancer. This was interesting because we'd previously found that CXCR2 could indeed influence chemotherapy responses in prostate cancer in a collaboration we'd had with David Warr probably about 10 years previously. But in vivo, we were never really convinced that the results that you would see would translate through to, you know, a positive clinical benefit if you went to a phase 2 clinical trial. Andrea's data again showed in genetically engineered models, or genetically derived models, as well as in standard human xenografted models, that actually CXCR2 inhibitors were able to reverse the progression of prostate cancer. So when you start to look at castrate-resistant prostate cancer, you can see that the CXCR2 inhibitor, here, the anti-CXCR2 antibody here, was able to influence the progression of in the TRAMP model, whereas the CSF1 inhibitor shown here was unable to do the same. He also went on to show that this was true for other subcutaneous mouse models, establishing a role for CXCR2 in prostate cancer, and actually went on then to demonstrate that actually CXCR2 inhibition could reverse tumors that were undergoing androgen receptor progression through an IL-23 mechanism. For the CXCR2 biology that we had for AZD5069, that left us in a position where we had insights to disease segments that we felt would be important to follow. So we could see that CXCR2 had a role in liver mets if we put together our liver met data and our liver met data from pancreas and our colon met data. That resulted in a trial Everest, which I'll talk a little bit about in the next few slides, and a trial that we then initiated from some subsequent work that was done to look at the role in the liver microenvironment for HCC, where we found again CXCR2 was controlling the immune microenvironment in liver cancer. And then also another trial where we looked at the reversal of androgen receptor resistance in prostate cancer that was published in Nature Medicine, I think, the year before last. And I'll talk a little bit about that. So for the early clinical development of 5069, in pancreatic cancer, we did 2 trials. We did Everest, which was supported by strong preclinical data. But rather than do a chemo-IO myeloid depleting regimen, we only did a combination with durva, and we did this in a second-line metastatic setting. The dose we took was 80 mg, and the size of the patient cohort that we were able to test was 20. So we were looking to see a clinical signal in a very refractory subset of pancreatic cancer, and in the absence of chemotherapy. The second place that we did a study was in head and neck cancer, where we didn't have any preclinical data for this. This was done as a comparative study versus an agent I'll talk about later on, which is the STAT3 ASO 9150, where we wanted to get data where we understood the comparative impact in 2 different tumor types, to sort of learn about this whole idea about disease-specific functions for myeloid cells. And again, that was done at these doses, and again, we had these numbers of patients, 20 to 30 patients to look at the signal. So within SCORES, we were able to build confidence that 5069 was a drug. As you can see here in the peripheral blood, the 5069 compound did exactly what it should do, which is across the range of patients and across both doses that we tested, we could see a rise in peripheral blood cytokines that bound to CXCR2. So IL-8 and the GROS, ENA78, were all upregulated in a dose-dependent manner and a time exposure-dependent manner when you treated patients with 5069. What we could also see in these patients was one of the things that's really limited the development of CXCR2 inhibitors was a depletion of neutrophils in the peripheral blood. So this is real-world data of these head and neck cancer patients. This has always been an issue because this is nowadays we would interpret this as mechanism-related PD. Yeah, in the past it was people were concerned about this because it was associated with a dose-limited toxicity that would potentially lead to febrile neutropenia. But this gave us some confidence that the thing was doing what it should do, that we're inhibiting neutrophil egress to the blood, but actually raised another concern, which is if you just look at the spread of the data on these plots, which is that the effect that it's having in the peripheral blood is variable over dose and over time. What we did notice in those plots was that it took a little bit of a while for the neutrophils to deplete in the peripheral blood, whereas the preclinical data would suggest that it would take some time. It shouldn't take any time for the neutrophil egress from the bone marrow to be evident. So we went back and we looked quite carefully at the time course of neutrophil depletion with 5069. So you can see here, this is in black, you can see the concentration of compound in the peripheral blood, and in blue, you can see the concentration of neutrophils. And indeed, 5069 does reduce very rapidly after the first dose of compound, the amount of neutrophils in the peripheral blood in an exposure-dependent manner. And if you look later, you can see that this is maintained. So there's no reciprocal rebound of neutrophils in the peripheral blood over time, or certainly over a 5-day dosing period. But what we did notice was very interesting is that if you looked at day 1, despite the neutrophils being down in the peripheral blood, the neutrophils in the tumor were not really heavily impacted. You had a small tweak at 6 hours, but these rebounded quite quickly. But at day 5, this is even in simple syngeneic models, you could see that then you were starting to get a sustained decrease in neutrophils in the peripheral blood, in the tumor microenvironment. So this led us to start to think about the fact that the timing and duration of exposure of the compounds was going to be important, and to understand whether to make sure that you'd modulated the tumor microenvironment ahead of introducing, say, chemotherapy or a checkpoint inhibitor. So that was all data that we were generating in parallel with the clinical trial data, because obviously we were responding to the clinical trial data at the time. We then went back and looked at how does this look in models of refractory disease. So in the KPC model, we looked at blood, and actually it was really interesting. In the blood, in the KPC model, we saw no modulation of neutrophil function when these animals were exposed to 5069. However, we did see a reduction, a very robust reduction in the number of neutrophils in the tumor microenvironment. So while the syngeneic model was telling us one piece of information, what we found in the disease-specific model, that we were getting a completely different read. And actually, this is interesting because in pancreas cancer, you've also got a much higher number of patients who exhibit a high neutrophil to T cell ratio. So it may well be that these disease-specific models have given us a much clearer read on the biology than we thought we would see before. In contrast, in the KPM model, again, a model where we've got liver metastasis, and we're driving effects, we saw a different regulation. So these high tumor burden, you can see that the CXCR2 small molecule was depleting neutrophils in the peripheral blood, and you see the same depletion of neutrophils in the tumor. So again, a piece of information that tells us that the way the compound was behaving, if you go from a syngeneic model to pancreatic cancer models to colorectal cancer models in situ, the biology is completely different between each of those different scenarios. We took a lot of that information to help design the ACE trial, which was run by Johan de Bono. What he did there was to then start to think about exposure and time of exposure within his dose escalation in that study. And so there is a drug-drug interaction with enzalutamide that he used to combine with AZD5069. So these the peripheral neutrophil control that you achieved was increased. So as you shifted from the 120 to 160 up to 320 mg BD, then you started to get really good control on the peripheral blood neutrophils, with a piece of information that we had late in the program that said, well, maybe what you want to do is to push the dose of these higher. And actually the fact that this has been tested and was well tolerated at these doses suggested that this concern around a DLT of neutropenia is really more about pharmacodynamics than it is about a toxicity concern. That's important learning for going forward with any neutrophil depleting therapy. And what was exciting with Johann's study was that he was able to see responses. So actually, he was able to see reversal of enzalutamide resistance, which is probably one of the first times that we'd really seen convincing data that translated from preclinical to clinical with a neutrophil modulator. And obviously this data is presented was published in Nature, which people can go back and read. But we were really, quite excited by this data and the ability to translate from disease-specific preclinical models to the clinic. He went on to do an awful lot more translational biology, and he could show that actually the types of responses that he was seeing, although it wasn't very clear what exactly the right dose was, it was partially dose dependent. Yeah, and that actually the responses that he was seeing was associated with good responses here, partial responses in drops in the peripheral blood neutrophils, were all linked together. So the peripheral blood biomarker data and the exposure data and the response data that he saw in the clinic, all linked together with the mode of action of the drug. So that really sort of led us back to a reanalysis of, what did we think about 5069 from a program perspective, and it fell into really, three major buckets. One, well, did we have the right patients? Did we have the right dose? And did we have the right combinations? So we think about the right patient. So in Everest, we think we probably at the time had the right approach, but when we look back, what we probably should have done is done a chemotherapy, neutrophil depletion, and an IO combination. And probably, it's very difficult to show that signal, a convincing signal, in the types of second-line patients that we were going into in that setting. The other thing that we probably would learn from that would be, to select which patients matched more faithfully the patients that we saw in the KPC model. Yeah, and one thing that we learned subsequently to execute in Everest is that CDKN2A mutations, which are quite common in pancreatic cancer, are associated with reduced response to IO. So actually there was a whole bunch of data that we didn't know at the time that would have refined exactly how we executed that trial. Head and neck for scores, we had no strong preclinical data to go into for head and neck due to a lack of availability of models. And in reality, the data that we had in scores, which I'll come and show you a little bit more in a few slides' time, sort of devalidated the role of neutrophils in that setting. And then prostate cancer, I've just talked to you, we did feel that study that Johann executed did validate the preclinical findings and generated some promising signals that provide us with good learning. What we didn't know at the time is the dose, and I think this is probably true for many myeloid treatments, particularly ones targeting neutrophils. We always have this concern about toxicity, and it may well be that actually we could have pushed harder with the dose. And then in the right combination, coming back to Everest, clearly 6CR2 biology together with a checkpoint inhibitor in a late stage setting needs a chemotherapy to give it some help. And actually, if going back again, looking at a more comprehensive therapeutic regimen would be helpful. We do have some exciting data that will be presented at a conference soon from Jeff Evans and the team up in Newcastle and Glasgow on the Cubic trial in HCC, where they're seeing some really interesting biology within the context of NASH. And again, another trial that we've done based upon the preclinical data that we've generated in more advanced models. And before we move off 6CR2, one thing that I do think is interesting is, how can we go back and use simple signals that we have from disease cohorts to refine patient assessments. These are two papers that I think are incredibly interesting, and actually if we'd had them before we started some of the CXCR2 programs, we would have used much more faithfully to guide how we selected patients and which disease segments we wanted to test the biology in. Just to give you some insight into the time frame over which this CXCR2 journey has evolved. So I started working on the project in 2001. It was the first project I was given when I came to AstraZeneca as an angiogenesis project, which we devalidated. And then we followed the science actually of this molecule all the way through to actually learning how to start to ask questions in the right way over about a 14-year journey, primarily driven in collaboration with the teams that put the beats in. But I hope this gives you a sense of the timeframes over which we're trying to evolve these science platforms. I'm now going to switch paces and pick up the pace a little bit to talk about some of the other programs we've had. So one thing that we invested in the PI3 kinase gamma program that was triggered obviously off the back of Judith's work in this space, but also a serendipitous observation around STAT3 was, how can we complement our neutrophil depleters and our macrophage depleters by reprogramming the macrophage and the MDSC populations. And these are two programs that we invested in, so STAT and gamma as two critical modulators of macrophage differentiation. So our drug in this to target STAT3 was an ASO, and this is a drug called AZD9150 that we developed in collaboration with an external ASO company. And we noticed something interesting in the phase 1 clinical trials in leukemia, which is that the ASO treatment was associated with the rise in an interferon signature. Yeah. We followed upon this sort of small piece of serendipitous data to see whether the ASO had the potential to combine with checkpoint inhibitors. And we found actually that pre-dosing the ASO and reprogramming, preloading the animals by depleting STAT3 did indeed enhance both PD-L1 and CTLA-4 responses, and that this was actually more effective than combining with our JAK inhibitor for AZD4205. So we found that this is an interesting piece of biology. We didn't know how it worked at the time, but we felt that it was worth pursuing based on that interferon signature and this preliminary data in the syngeneic models, and that we could demonstrate that this was through a CD8 mediated mechanism. We went on to look at where was the STAT3 ASO going, and what we actually found was that the STAT3, perhaps not a surprise, we should have thought about it ahead of time, was not really targeting the tumor cells that we thought we were targeting with it. Here's the cancer cell uptake. But what we were seeing was a significant uptake of ASO, sorry, the bottom, ASO into the myeloid populations, and in particular into the macrophages. Yeah, and it's in these cells that we could see the most significant downregulation of STAT3 expression. So we exploited that observation to drive the development programs of STAT3, and that's what resulted in the trial that we did in SCORES. But we also found some really interesting things around the STAT3 ASO. So one thing that we did find was that this was differentiated from both CXCR2 inhibitors and CSF1 inhibitors. So one thing that we were looking at was resistance to STK11 mediated CPI resistance. So we generated a suite of models in the CT26 where we'd knocked out STK11 and shown that these were, although sensitive and as a wild type to double checkpoint inhibitors, they were completely resistant to double checkpoint inhibition when you'd knocked out STK11. And what we found was that STAT3 was really good at repriming the IO response in these STK11 resistant models shown here on the right. But what was really interesting was that they were not, the STAT3 ASO was doing something very different from both the CXCR2 inhibitor and the CSF1R inhibitor. And this sort of gave us some hope that actually with CXCR2 and with CSF1R and with the STAT3, potentially with PI3 kinase gamma, which I'll talk about in a minute, that we were able to deliver different phenotypes to patients to modulate the microenvironment in different ways. I mentioned scores before. This is the phase 2 clinical trial where we tested the STAT3 ASO and the CXCR2 inhibitor head-to-head in both PDL1 naive and PDX naive and PDX pre-treated patients, combining with checkpoint inhibitors with durvalumab. What was interesting in this clinical trial is that we did see some signals of response. So the STAT3 ASO gave a small number of complete responses and a small number of partial responses out of the roughly 40 patients that were treated in combination, whereas the CXCR2 inhibitors basically showed no responses. And so this is maybe an indication of a phase 2 clinical signal for this asset, but it was limited by the standard ASO tox of thrombocytopenia, and obviously CXCR2 inhibitor was associated with neutropenia. However, one of the things that concerned us was that when we looked at the pharmacodynamics and looked at how much STAT3 knockdown we were able to achieve with this mechanism, despite the fact that there was some very compelling preclinical biology and some interesting clinical results, the degree of STAT3 knockdown that we could achieve by mRNA analysis of peripheral blood was not at the level that we felt was sufficient to deliver a robust regulation of STAT3 in a broader population. So we really had quite a lot of learning from this program. So although we did park it, it wasn't unsuccessful. This ASO approach did give us an opportunity to enrich for STAT3 knockdown in macrophages and MDSCs. It was able to enhance the CPI with different mechanisms, and there was evidence of favorable PD and efficacy in the clinic, although the dose that we were able to see did not warrant moving forward to sort of phase 2b or phase 3 clinical trials. One of the things that may well have been interesting for this asset is that maybe patient selection may have increased the magnitude of benefit that we saw, and gave us more confidence that the degree of STAT3 modulation that we were seeing was sufficient. But it still led us with the open question, do we need a higher degree of STAT3 regulation to recapitulate this mechanism with another therapeutic modality? Obviously, people are very familiar with the other approach to modulate PI3 kinase, modulate macrophage modulation, which is PI3 kinase gamma. Obviously, these are Judith's papers on the left and the partner paper from Dehano that were published in Nature. We had a very similar molecule, which is shown on the right, which effectively phenocopied IP549. That was originally developed much like our CXCR2 inhibitor for asthma and inflammation. And in fact, this data here shows how well that compound modulates neutrophil function in peripheral blood, which is not something that was really been exploited in a cancer setting. This is the comparative data that shows how well it mimics IP549, where we can see in a whole load of primary cell assays how comparable 358 and 549 are in terms of their preclinical profile. And again, it shows exactly the right profiles for modulating macrophage differentiation ex vivo in many different types of macrophage differentiation assays. The thing that was really interesting about 3548 was that, again, much like the STAT3 ASO, it had the ability, much in a recapitulating Judith's work, that it was able to enhance PD1 responses, and it had exactly the right type of phenotypic responses in the syngeneic models. But again, very much like the STAT3 ASO, in this STK11 depleted double checkpoint resistance model, where the checkpoint resistance is in the absence of chemotherapy, again, you could see that the PI3 kinase gamma inhibitor, as a reprogrammer, was able to enhance survival benefit in clearly refractory tumor models. So again, macrophage reprogramming with STAT3 and PI3 kinase gamma was able to reprogram myeloid cells in the context of STK11 loss, whereas depleting cell neutrophils or macrophages, or even combined inhibition of CXCR2 and CSF1R was unable to recapitulate that biology. So clearly reprogram is different from depletion, and the setting where that is most efficacious is going to vary between potentially tumor mutation statuses. So from our 358, for our PI3 kinase gamma efforts, we saw really promising preclinical activity in the models. We were able to generate a compound that was similar to eganelisib. And we had a setting, the STK11 setting, where we felt that probably the biology that this could deliver was different from what we could achieve with a CXCR2 or a macrophage depleter. However, one of the things that was obviously we were keeping a really clear eye on was the success of eganelisib. This is, I still feel as though the biology that's been delivered with that compound in the clinic is really key for the myeloid field. The data in the Mario trials was promising, and the combination that compound was able to deliver with chemo and checkpoint inhibitors, again, I still think, even though they're small and they have not been recapitulated in big phase 2Bs or 3s, they're still exciting and point to a really exciting potential still for these types of agents in the clinical setting. One of the things that we did invest a lot in as an organization was metabolism. And in the myeloid field, we looked at arginase, like many others. For those not familiar with arginase biology, arginase catalyzes the conversion of arginine to ornithine and urea. But arginine in the peripheral blood is required for optimal T cell function. So myeloid cells expressing and secreting arginase can result in immune suppression within the tumor microenvironment. So the approach that we took was to look at how we may approach the arginase question in a slightly different way than the competitor molecules that were being developed at the time, Trincalathera and well, these two molecules here, which targeted intracellular and extracellular arginase. We were concerned about the role that arginase plays in the liver, and then being limited by liver tox clinically. What we did was we developed AZD0011 as a candidate drug where it only inhibited extracellular arginase. Originally, we developed it as a prodrug so that it would drive absorption through the gut. And we found that the actual active drug had poor cell absorption. And so effectively, it was only inhibiting arginase that was present in the peripheral blood. Despite that, this arginase inhibitor was able to deliver anti-tumor immune enhancement in combination with PD-L1 inhibitors across a range of syngeneic models. When we did the profiling of these models alone and in combination, what we could see was that the arginase inhibition of secreted arginase was enhancing the ability of checkpoint inhibitors to drive an optimal immune response within these syngeneic models. So this taught us one thing, which is that actually maybe we shouldn't be making an assumption that you need to inhibit arginase or other drivers of immunometabolism suppression in the cell, intracellularly and extracellularly, and maybe there are ways in which we could get to inhibit the biology in a much more compartment specific manner, maybe to drive greater inhibition. And what we were able to show is that this profile could deliver an arginase inhibition that was differentiated from the other inhibitors that were targeting intracellular arginase. But one of the reasons why we didn't progress with this program was that we felt that there were things that at the time that we didn't understand. So what is the pivotal immunometabolite in a given tumor setting? What's the balance between arginine and other things we know are important, such as glutamine loss or high lactate, adenosine depletion, the levels of kynurenine? And at the time, the kynurenine IDO trials had failed, and that sort of put much more pressure on these questions from a development perspective. And really, how does the metabolite profile change when one pathway is inhibited? Do the other immunosuppressive metabolites take over as a suppression mechanism? So we have still retained an interest in this space from an understanding perspective. And we've been investing using spatial omics to look at the immunometabolic map of both simple syngeneic models, and here shown a suite of preclinical models where we're using mass spec imaging to look at the spatial distribution of these immunosuppressive metabolites. This is work that we did as part of a CRUK Grand Challenge program, where we've started to look at how does adenosine respond as a primary suppressive feature, but also how do these other immunometabolites look within the context of a complex tumor microenvironment. And this is work that we're continuing to invest in to understand the importance of metabolism, how it influences immune cell behavior. Well, one of the things that we were really quite interested in is studies like this that have started to come out, and this was recently published, where we're seeing things that are derived from macrophages like itaconate that are influencing tumor cell behavior. And, you know, metabolites like this, I think, are going to be really quite interesting if we can harness them in the right way. We're starting to refine how we can tackle the immunometabolism space and the tumor metabolism space, and we're starting to generate better inhibitors that will allow us to tackle biology such as arginase, lactate, and now the evolving role for itaconate and the glutamine-glutamate ratios. And these could offer really interesting approaches to look at that interaction between the myeloid cell compartment and the tumor cell compartment. And then lastly, I'm going to very quickly whiz through our work in the macrophage depleter or recruitment space. So this is a really something that we really struggled with for quite some time. Is, you know, why did the CSF1 inhibitors not give us the effects that we were expecting to see in the clinic? And we had a whole bunch of questions that we decided again to go back to the GEMS models and look at, particularly in the context of colon, where we know that there are subsets of colon that were significantly enriched for macrophages. And this is work that was done by a postdoc up at the Beatson, Nohar, SM Mohammed, where she took colon tumors, and what she did was she took a slightly different approach, which, knowing that we could align the genetic models to disease subsets CMS1 through 4, she then went back and classified colon cancer for their macrophage phenotypes within the context of the CMS1 through 4 segments. And using this information, then we were able to look at what the macrophages were, what phenotypes they were, and then understand, if we looked at models representative of these different subsets, how they would respond to CSF1R inhibitors, CCR2 inhibitors, or how would tumors progress in models where we're deleted for the CCR family of 1, 2, 4, and 5. So the first thing that we tried was the metastatic model that we knew was sensitive to neutrophils, so the metastasis model, the KPM model, and also a similar model of CMS4 AKP. What we found here was that both depletion of the CCR receptors and prevention of macrophage recruitment in that way had no effect on progression of models, these two different types of models in the CMS4 segment, nor did CSF1 dependent macrophage depletion have any impact on these two CMS4 tumors. We also found that models that were reflective of CMS2 and 3 did not show any benefit to CCR or CSF1R mediated interventions. But what we did find was that in models that were reflective of the CMS1 segment, that we saw an increase in a clear increase in preclinical benefit when we depleted CCRs, but we saw no benefit or very little benefit, even detrimental benefit when we depleted CSF-dependent macrophages on background of CCR knockout. So what we found was that actually, if you're looking at macrophage biology, what was really important was the subset, and the subset of colon that appears to be dependent, at least preclinically, on macrophages, and also has high T cell content, is the CMS1 segment. But the segments that we primarily focus on is, you know, as in 2, 3, and 4, don't appear to be influenced by that. Moreover, that actually the CSF1R mediated neutrophil macrophage depletion could be detrimental on the back of inhibiting the CCR dependent population recruitment to the tumor. So again, Not all the mechanisms are the same, but actually it leads us into understanding that there are segments that we could go into to gain confidence that our myeloid modulating therapies are working and are leading us to a subset that we can identify and select in the clinic. This idea that CSF1R is not depletion may not be helpful in all settings is also recapitulated in a time-dependent study that was published a couple of years ago from a collaboration with Fran Bolkwell and Owen Heath, where they found that actually if you treat animals with CSF1R inhibitors, actually you can abrogate, and in fact it negatively affects the response to carboplatin in ovarian and mentor models. So we know that there's actually a time-dependent role for CSF1 dependent macrophages in the response to platinum treatment within colon cancer, which is consistent with the buildup of macrophage pools following platinum-based treatment of humans and in the preclinical models. So, you know, what we are trying to do here is to learn a little bit more about where the cells are important and where there are positive and negative effects of the inhibitors, to understand how we should schedule and sequence the models, the inhibitor administration in combination with both platinum and IO. Okay, so those are that's a sort of a quick whistle-top tour around the biology. What has it taught us? So something that was really important within the context of AstraZeneca is this concept of right target, right tissue, safety, the right patients. And there are a number of places where the data that I've tried to show you today has been targeted to allow us to understand, you know, where should we be, which target should we be taking, myeloid target should we take into different diseases, which tissues, so primary tumor versus secondary tissue, should we be, this is metastatic tissue, should we be focusing on, and with what exposure, and then what exposure can we deliver to the patient in the right combination potential to understand the safety. So really, you know, how do we refine our treatment regimens to drive better success in the clinic? Why is that important? Is that something that we invest a lot of time on, and is understanding the relationship between dose time, frequency of dosing, to understand how we are driving the biological effect, target suppression, pathway suppression, and outcome, and balancing that out off versus the therapeutic index. So the PD in normal tissues, the tolerability that you see in the overall patient, and the efficacy you can then drive with a given combination. And really trying to understand where do we need to pitch a treatment or select different types of treatment to get the right balance between toxicity and target engagement. Everything that we do and are now thinking about in the context of, you know, things like the myeloid treatments is focused on getting this schedule, sequence, and combination potential better in the context of defined patient segments where we're selecting the combination partners in the most appropriate manner. The second thing that we're thinking really hard about is where do these agents sit within the context of these increasingly complex tumor microenvironments. If we're able to focus the type of information that we now have in the, you know, in all of the really elegant papers that have been coming out in the last few years around the complexity of TME, can we start to think about simple biomarkers that would allow us to get confidence, generate confident phase 2 clinical data where we're shifting survival curves in the way that people expect to see them, where we're seeing clear daylight between our backbone therapy and then the enhancement efficacy that we see with our myeloid therapy. And what's really important is that, why is that important? Because we have 20 to 40 patients. I've tried to show you in some of the clinical trials, we have 20 to 40 patients roughly to show the clinical benefit. Yeah, so if you don't select, and that we're trying to explain the biology that we see in the context of an increasingly complex and unrefined patient cohort, then generating data that looks like this, which is what we want to see for ongoing investment towards phase 3, is going to remain difficult to generate. This is a couple of slides I'm going to finish on from spatial biology work that we do. So the field is really in the same space. we're generating a huge volume of increasingly complex data as we move to single cell, in situ, and spatial approaches within the myeloid field and in the TME field in general. One of the things that's really important for us from a drug development perspective is to use those type of data as a sandbox for learning, which is where I think we are as a field right now. But what we need to really focus on is how we then refine those biomarker information and biology information down to generate biomarkers that we can deploy for phase 2 and phase 3 patient selection. So a small number of biomarkers that are robust and cheap that we can deploy across clinical centers to enhance the refinement of our clinical trials. And that plays into my last slide, which is, what have I learned over the course of executing these different programs as being important? Mechanism, combination, selection on the left, and putting together the right combinations to drive an optimal T-cell response. And on the right, thinking much more about dose, sequence, and schedule to optimize the way the patient is receiving the drug. And then finally, the acknowledgements, all the work that we've done has been in collaboration. We've done very little of this work in-house, apart from the work in the syngeneic models. And, there's a lot of people from Glasgow and from Andrea's group in particular that we're very grateful for who've really spent a good 10 to 15 years working with us on the programs. And with that, I'll stop.
Okay. Thank you, Simon, very much. We will invite people to ask. We have quite a few already in the chat, so I'm pretty sure Judy wants to ask, but I will keep her on the dessert. We will start with questions in the chat. So the first question, a little bit misunderstood, but maybe it will be clear. Have you explored the role of pan-MDAC, I guess all MDAC, and development of NEPC? NEPC.
No. I think that the majority of work that we've done, well, unless you have, obviously, but the majority of work you've done from a small molecule perspective has been on the programs that I showed you. So really the classical mechanisms of regulating myeloid cell recruitment or function within the tumor microenvironment. Yeah, and largely the reason for that is that those targets are tractable to us, so we can develop drug programs against them. Yeah, whereas to target some other mechanisms is a lot more challenging.
All right. Another question. With arginase inhibition, do you see increased nitric oxide in the microenvironment and blood? Does it affect blood pressure?
Yeah, that's a good question. In the non-clinical models, we didn't. Yeah, but what isn't really clear is whether that's true for the two pan-arginases, the intracellular and extracellular arginase inhibitors that have gone to phase 2 clinical trials. So some of those are ongoing, but I'm not sure that data has been reported yet.
Okay, next one. Great talk. Question. Given the rise in IL-8 with the CXCR2 inhibition, would there be benefit from dually targeting CXCR1 and 2? It's from Rodipina. So he said that we've used the CXCR1 and 2 inhibitor across various models with a significant anti-tumor activity in PDAC, colon cancer, and so on.
Yeah, so I think that's a really interesting inhibitor. Yeah, so I would say that the CXCR1 and 2 mixed inhibitor is a really interesting asset, and it would be I would be quite excited to see how that biology could be progressed. So I think that could be something that you would take on from the CXCR2 selectives to drive a more comprehensive neutrophil modulation. I think the only thing that I would probably say with that is to get high enough up the dose in the clinic, yeah, to get the type of modulation of the TME that we'd want, learning from 5069, you may want, we probably have to think about intermittent dosing.
Okay. Another question. Have you investigated the arginine in TME generated by cancer cells?
Yes. So we have looked at that, and that was one of the triggers for doing this comprehensive immunometabolism map. Yeah, so I can't quite remember the exact distribution of the data across the different models, but we will be publishing hopefully sometime late this year or next year a map of the arginine distribution.
Next question on Dan Watterson. Were there difference in the amount of STAT3 knockdown observed in respondent versus non-responder patient?
That's a really good question. We didn't have that many tumor biopsies, so all the data that we could generate on the STAT3 knockdown was by signature, RNA signature in the patients, and that wasn't differentiated by response.
That's next question is from academic researchers, I guess, and I will tell you why I'm smiling. Can you provide information on availability of some drugs, MCFR1, STAT3 inhibitors for preclinical studies for research outside AstraZeneca?
So the CSF1 inhibitor, we have, AZ has an open innovation website, which we can provide, the way you can put in a request to get access to the AZDs. The STAT3 inhibitor we've now handed that back to Ionis, and you would have to contact Ionis for the STAT3 inhibitor, but the other drugs, they can be available through an MTA process.
Yeah, and CXCR2 as well, right?
Yeah.
Okay, next question. Thank you for such a nice accumulation of impressive work. Great. Did you consider testing CXCR2 inhibitors in other diseases, non-cancer, such as ANCA, vasculitis, ischemia reperfusion, right?
We didn't do ischemia reperfusion, but one of the things, I guess I'm not a very patient person, but one of the things I had to be was patient. So the CXCR2 inhibitors within AstraZeneca were originally developed for COPD and asthma, and people are never keen to do trials in both COPD and asthma and oncology because there's different criteria associated with those disease settings. So I did have to wait a significant amount of time to get hold of my 5069 because that was the best in class CXCR2 inhibitor at the time. It was efficacy. It did show disease modulation in COPD and asthma in terms of neutrophil flux to tissues, but a degree of disease modification that it delivered wasn't sufficient at the doses that were administered.
And this is actually the last question, but it's very related to the previous one. Have you used macrophage recruitment drugs outside the context of cancer? I blocking recruitment.
No, I haven't.
CSF1R, right? You probably
no, we haven't. Yeah. Within other parts of the business, obviously the PI3 kinase gamma, that has been looked at within the respiratory and inflammation settings. That's with that biology originated.
Well, I guess Syntax approved their drug for the GVHD, right? Which is kind of cancer, but it may be considered non-cancer, right? Indication as well.
Yeah, and I think that's really quite interesting. That whole route forward is quite interesting.
Well, it's a great idea that just some drugs which has a tough line in one type of disease can be useful in other, and that's good.
I'm still hopeful that eganelisib though is gonna, someone's gonna turn that into a myeloid modulator.
Yes.
Yeah.
This is the last question on chat. Is anybody wants to ask questions in the microphone, please? Judy.
I do. Yeah. So did you ever observe any effect on vascular leak when you studied your PI3 kinase gamma inhibitor? One of the lesser known facts about PI3 kinase gamma is that it regulates endothelial cell-to-cell junctions, and when it's activated, it actually causes vascular leak, and inhibitors of that are really, really effective at blocking things like vascular leak in sepsis models and in
no, I mean that's interesting, right? Because that's no, we didn't, right? That's an ignorance on our part. But that's sort of intriguing because if you think about the whole VEGF thing and the lower dose VEGF priming, T cell rolling and recruitment,
Mhm.
not through the angiogenesis thing, but just about making sure that the vessels are competent to recruit T cells. That might explain in part why gamma and STAT are different from CXCR2 and CSF1R.
Yeah.
That's
Yeah.
quite exciting. Yeah, that could be quite cool.
Yeah. There are a lot of diseases where vascular leak is a major issue. In fact, the TG100-115 compound that was originally developed here in San Diego was developed for vascular leak indications before we started using it in cancer models. So, yeah. So I had a question about whether you ever combined PI3 kinase gamma inhibitors with CXCR1 inhibitors. Did you ever think about that?
With the two.
Or two, yeah.
Did we do that?
Mhm.
We did it in some experiments, but largely as a negative control. Not negative, I mean, like yeah, we did it as large as a negative control. But we didn't really ever see. It's something that confuses me, right, about the PI3 kinase gamma biology is that we developed those inhibitors as neutrophil modulators originally within AZ. Yeah. And all the biology was driven by neutrophil. And then but we see a different effect in a CXCR2 and a gamma setting. That's something that to this day I still can't get my head around.
One of the questions I wanted to ask is, okay, in some tumor models, when you inhibit myeloid regulators, you actually get an increase in pro-inflammatory myeloid cells, and you also using some other therapeutics. So sometimes inhibiting myeloid cells is beneficial, and sometimes the treatment can recruit more that are actually beneficial. So have you ever observed that, and do you know work along those lines?
Yeah, so we haven't looked at it as an opportunity to drive more in. I haven't from a small molecule perspective, but what the aspect that we've looked at it from is what I try to highlight, which is a dose schedule sequence. So, if you deplete them for too long, do you get an acute benefit from the modulation of the TME, and then a negative benefit because you haven't left the normal cells to mature into the tumor in the right way to aid the progression of the immune response, or T cell cycling or whatever else is going on. So, for example, the work that we did with Fran Bolkowicz, I highlighted the Owen Heath model in the Amentum platinum response in ovarian cancer. At the time that was quite perplexing because he said, well, I've actually got these macrophage pools that are associated with positive response to platinum in ovarian cancer. And then he showed very clearly that there was a time dependent, if you took out the CSF1 dependent macrophages, you attenuated that response. So I'm really very conscious in my own mind that, if you suppress neutrophils chronically or you deplete macrophages chronically, then you may well give with one hand and take away with the other. It'd be like the tax man, right?
Yeah.
Yeah. And I
I
think
think that's what
something
makes the
is
field
important.
so difficult.
Yeah. Yeah. And I think, and that's why maybe, things like eganelisib are quite exciting because you change the differentiation state. You may not need to worry about this on and off, up and down, remodeling and timing issue as much with those types of therapies as you do with others.
Yeah, very interesting.
Okay, do we have any more questions from people? We still have quite a few people on the call. Before I close it, going once, going twice. Okay, Simon, thank you very much. It was great. We're already 10 minutes past the time. Thank you everyone for attending, and see you next month. Thank you.
Thanks, everyone. Thanks,
Thank you,
Dimitri.
Simon. Bye.
Judy, I think we can stay on this link, Dimitri, Jelani.
You want us to stay?
Well, this is the same link we used to meet, so.
Right, I'm staying. I didn't know that we were supposed to stay. Okay, I can do it.
I usually just kick off people who